Namaste and good morning, everyone. I heartily welcome you to all to the International Symposium on Emerging Trends and Healthcare, jointly organized by Center at Research and Innovation and Translation at Acme University and University of Miyazaki, Japan. Currently, we are waiting for our dignitaries. Kindly keep your patience. We'll be starting our inauguration soon. I would like to request everyone in the audience to be poised. You can wear your iCards, please. Yes. And kindly turn off your phone or silent them. Kindly wear your iCards and silent your phones. Yeah. Namaste and good morning everyone. I heartily welcome all of you to the International Symposium on Emerging Trends and Healthcare, jointly organized by CRIT Center for Research, Innovation and Translation at Atmi University and University of Miyazaki, Japan. Today we have with us our esteemed guests, Dr. Harish Kumar Madhyastha. He is professor at Miyazaki University, Japan, and Dr. Manas Kumar Santra. He is currently scientist F at NCCS, Pune. I would like to request both our dignitaries to take their seat on the dais. Along with them, I would like to request Dr. Ashish Kothari sir, director at RIT AU, to kindly take his seat on the dais. Ravi Tak sir. I think we are currently waiting for Ravi Tak sir, right? Okay. I would like to request Dr. Krishna Joshi, HOD of Microbiology Department at Atmi University, to take the seat on the dais. We have a tradition of chanting 3 Omkars before continuing any pious beginning of any event. Let us gather for Om chanting. I'll chant from here. Kindly join me. Now let us pray to the lotus feet of Thakurji. Kindly prepare yourself for prayer. Now I request all the dignitaries for garlanding the feet of Thakurji. Kindly, sir. Participants, please settle down. Okay, we move forward to our next step of inauguration, that is lighting the lamp. Any good deed blessed by Lord Agni is also a success. So I would again like to request all the dignitaries on the dais to please enlighten the lamps. I would request Dr. Maushmi Das and Dr. Shivani Tak to join there, as they are coordinators of this event. Now let us move forward with our next session of inauguration, that is formal welcome of all the dignitaries on the dais. I would like to request Dr. Ashish Kothari sir to welcome Dr. Harish Kumar Madhyastha as a formal welcome. I request Dr. Bhargav Waghela, Assistant Professor at Department of Microbiology, to welcome Dr. Manas Sandra. I request Jinesh sir to formally welcome our Registrar Dr. Ashish Kothari. Now I request Dr. Maushmi Das, our coordinator of the event, to welcome Dr. Krishna Joshi. I would also like to formally welcome all the dignitaries of the dais, all the professors, and I would like to heartily welcome specially to all the participants whose enthusiasm is making this event a success. Now let us move forward about some introduction to this event. So this prestigious international symposium on emerging trends in healthcare is bringing together all the global experts, researchers and industry professionals to discuss this transformative innovations in healthcare. This international symposium has received 142 participants, among them which are 48 are posters. And as it is going to be a YouTube live session, it will be joined globally by many participants. So truly it is an international conference a symposium in every sense. Now I would like to move towards the end of the inauguration session, and I would like to request Dr. Maushami Das to provide vote of thanks.
Good morning, one and all. Thank you so much for the presence. In spite of busy schedule, especially the faculty members, the deans, HODs, and other respected dignitaries, especially our esteemed guests from Japan as well as from Pune. On the end of this particular inauguration session, I would like to thank all the participants who have participated from inter-university and intra-university level, as well as the very all the best to the poster presenters. And after that, I would also like to take the blessings from Param Puja Swamiji, and I like to start the expert talk session followed by this and hand over the mic again to our anchor, primary anchor. Thank you so much, and other participants, please attend the expert talk session. It will be most surprising to get new knowledges, new areas, dimensions, which will definitely give you a path break research innovations to follow in the next future life. Once again, welcome to the next series, expert sessions. I request dignitaries on the dais, please take the seat of the dais so that we can start the expert talk sessions. I request the staff support to release the chair, sofa and the tables so that we can start the poster presentation. Thank you. Volunteers, please come forward. शुरू में था ना, शुरू में दिख रहा था ना यूट्यूब में।
Let us move to our expert talk session. First of all, for today's first talk is of Dr. Manas Kumar Santra. Dr. Manas Kumar Santra is currently Scientist F position at NCCS Pune, India. His field of research currently is around cellular, molecular, and epigenetic aspects of cancer pathogenesis. He did his BSc, MSc, and in chemistry, and he did his MTech in material science from IIT Bombay, as well as he was awarded his PhD from IIT Bombay in structural biology, and he pursued his postdoc from Massachusetts Institutes of Technology, MIT, USA, in field of cancer research. He is NASC Fellow of Charles College King. Then he is receiver of many prestigious awards. He has, yeah, he has under his guidance 15 PhDs and 96 publications with more than 3,000 citations. I would like to request Dr. Manas to continue with his talk. Kindly welcome him with warm applause.
Point. Sure, sure, sure. Okay, thank you, madam, for nice introduction, and my sincere thank to the organizer for inviting me, and respected dignitaries and professors and my dear students. So today I'll talk about the life of protein. Okay, this is just think about that whether protein has a life. Okay, but my appeal to the student is that you stop me anywhere, anytime, if you have question, because that will give me the opportunity to learn more from you. Okay, so any moment of time, if you have any doubt, any question, just stop me there, we'll discuss, because that is the opportunity I want to have it to learn more from you. Okay, so how many of you think that protein has life? Okay, so before going to the life of protein, let me go to that first, talk about the life of ours. Next slide. See, this is, if you think about human life, okay, so we start from a single cell, fertilized egg, and then fetus, then baby, then child, then teenager, adult, old age, and the ultimate is death. Okay, do you think the protein has similar life cycle? Anybody of you think? Because, can you go to the next slide, please? Okay. How organism becomes multicellular. How? Can you go to that next? Okay. So the fertilized egg, you see that from the single cell that become 2 cell, 2 to 4, 4 to 8, 8 to 16, and so on, and then ultimately that birth happen. Can you get the next slide, please? Okay. How it happens? How this single cell to 2 cell, 2 to 4, 4 to 8, and so on, how it happens? This happens because of cell division. One cell become 2 cell become 4 cell. Okay, next slide. So what happens during the cell division? Actually, why it is happening, how it is happening. Okay, so you see the mother cell, that genome, okay, the genome, that where our all the informations are stored, that is the genome, which will be duplicated and then will be segregated and then will be separated, and then ultimately two daughter cells happens. And cell division is the process all about. And इसमें
आगे ये नीचे
हाँ,
ये टॉर्च ठीक है
okay.
टॉर्च का
Okay, fine.
ओके सर
Okay, thank you. So, basically, if you think about it, that this is the one step, and then the segregation happens, then equal separation happens, and then two daughter cells happen. And all our information is stored in this, in the form of genetics, but a chromosome in our case. So, what did, okay, see. So this is actually happen in the cyclic manner. Okay, it will happen, that is called that cell cycle. It happens all the cyclic manner. The cell growth happen in larger size. It is the metabolically highest active state. Then the stage where genome duplication happens. And if you know the genome duplication, if it is improper, so many diseases are associated with, like Down syndrome, okay, many, the cancer. Then another, the equal distribution. If this is the phase, it is happen. If equal distribution is not happening, then what will happen? Many disease, including like cancer, the aneuploidy, because one cells will get more number of genome, another cells will get less number of genome. So the aneuploidy, the cause of aneuploidy happens like this. And you know this phase, that one of the best, one of the most important cancer drug is used to disrupt this, to prevent this, that is that Taxol. Okay, so that's the cyclic way, the G1, S, G2 and M phase. This happens. If never precisely happens, then the disease called cancer will happen. Okay. So this is that's the one cell to become two cells. The cyclic way should happen, and it should happen in the precise manner. And all these works are done by basically proteins. So how protein, the constituent or the major constituent within the cells is the DNA, where our all information are stored, and that will be actually from that RNA through the transcription process. Okay, that should be also precise. Then RNA actually will be in the form of the protein will translate, and this is very important, because this protein, without protein, nothing work can be done, not a single work can be done within the cells. Now this protein, at this stage, after the newly synthesized, it may be functional, may be non-functional. Okay, there is the important steps happens which is not in any textbook that much. So that is called the post-translation modifications. So this kind of post-translation modifications will happen. So these are basically, I'll come to this, that what is the post-translation modification. They play very, very critical role whether proteins should function or do not function, or what kind of cells can, or what kind of proteins do what kind of function. Even same protein can function differently based on their type of post-translation modification. Same protein will have multiple role based on this kind of post-translation modification. So multitasking, the many protein are multitasking. They functions at the membrane, they functions in the cytoplasm, they function in the different organelle, they function in the genome. Okay, so based on basically what kind of modification the same protein is undergoing. Okay, it is basically context dependent functions will be controlled by entirely by post-translational modification. And you know the protein is the basically all functions need, within the cells carried by the protein. So protein is nothing but the polymer of amino acid. And many of you have already know that amino acid. So we have, we have 20 amino acid, and permutation combination of this 20 amino acid, we have proteins. Do you know how many proteins in each cell we have? Approximately how many proteins we have? Almost 34,000 proteins we have within each cell in our human body. Each cell which we cannot see our eye without microscope, there are so many proteins are there. And those proteins are coming because of permutation of combination of these 20 amino acids. So our basic amino acid, acidic amino acid, 2 amino acid, then that polar amino acid, and this is the special case. Why is special case? They are the helix breaker. Okay, this glycine and then that proline and then cysteine. This is the unique amino acid where covalent bond formation happen. Two proteins can form that bigger protein through covalent is basically that cysteine. Okay, many of you have heard about the basically antibody. Antibody has heavy chain and light chain. They forms the antibody through covalent interaction through the cysteine, and that is playing very important role the functions. And this is the hydrophobic. Okay, so hydrophobic amino acid plays critical role like membrane recruitment. Any protein, they will have the hydrophobic, they will be in the membrane. Okay. So then, do you believe that protein has a life cycle like us? Okay, actually understand that you'll see the surprising that like proteins will have this life cycle like us, that you know the protein synthesis happen in the ribosome. Ribosome will the surface of endoplasm. So there's the synthesis will happen, that is the born. Then brought up is the post-translational modification. Depending on the post-translational modification, they will be in the anywhere within the cells. So here we are showing that it is in the membrane. Okay, and once this function happens, functions is over, this protein will die through two important organelle. One is that proteasome, another is the lysosome. And as I said, within the cells, we have almost 34,000 proteins. Any cells, okay. And then you know that this is the proteasome, almost 35,000. And it is a kind of dustbin. You can think about it within the basically cells, there are the 35,000 dustbin. Okay. And in the lysosome, that is another kind of dustbin. Almost 1,000 lysosomes are there within the single cell. And if you actually, like, you know that we are in the city life, if our waste management is not proper, what kind of disaster we will have? The exactly same things, if you actually disrupt this dustbin, okay, the all protein will die, almost 80% protein will die in this organelle. This is called, you can say that it is a dustbin, or in our human things that kind of assumption. You can think about it. So basically that here it is playing critical role, and you know the one of the drug which actually inhibit the function of lysosome, and that is routinely used, means that first line of chemotherapy drug in the multiple myeloma. Multiple myeloma is one kind of blood cancer. So there the drug is used, bortezomib, that is the first line of therapy of multiple myeloma. That drug basically disrupts the function so that all the proteins will be chaos, there will be that no protein will die, nothing will happen, crowding, and the cells will go for die. Okay, so that's why that in our society, whatever that waste management system is very essential for that life. Similarly, cells has the same beautiful system, almost more than one dustbin for each protein. Okay, so that is the clinical use. So this is basically, if we think in much broader sense, you see that in the ribosome, the protein synthesis happens, and then separate protein comes, they will make the fold, this kind of folding happen, and then once it is properly folded, that is the baby happen. And then if we think about it, this protein undergoes series of different types of post-translation happens. This is kind of chilled, when hair growth, teeth, eye, everything that mature happens. So that's the basically maturation child stage is basically controlled by different types of post-translation modifications. Then when it become, then is the teenager. You see that basically two different proteins, protein-protein interaction, because that in the society we cannot live alone. We need neighbors, we need everything, society. Same thing that it happens, protein, they crosstalk each other to do some functions. So depending on the interact on this protein, for example, take this protein as example, it can function multiple ways depending on the whom it is interacting. Okay. If it interacts in the nucleus, it will function different. If it interacts in the membrane, it will function different. If it interacts in the mitochondria, it will function different. Depending on that who is, like just like father when he is in that with son, daughter, the different role, different function, when is a wife, when is a mother, like that way. You can think about it that the protein is actually like that kind of life it has. Then it is, when it is interacting much more protein, as I said, in different contexts, this is the adult stage of the protein. Okay, so it is multifunctional. Each protein, you can think about it that each protein of our body is doing the multifunctional. Okay, and that they do because of this interaction. And then when the protein, each life has that certain time, so same thing after this function, it will be marked for the cell, means protein death. That is the only post modification, that is the ubiquitination. I'll come in that. This, when the protein is marking at the end of this, end of their function, so this will be the marking is very, very important. So that means there is no more needed this protein in the cells, that is the old stage, and so then the death will happen. So I'm now basically, I think now you understand that the protein has similar life cycle like us, okay, and it is essential. That's why you see that different protein, like our human, that some of us die at early stage, some of us is middle, some of us actually complete 100 years. Okay. Similarly, protein has also similar life from 15 minutes to 24 hours. Okay, like structural protein, like basically the microtubule, actin, these are the proteins that or architecture protein like nucleus, architecture, so they have the long life like 24 hours and more than 24 hours. But many protein like cell cycle protein, they have 15 to 30 minutes like cyclins, one example. So they have very limited because they need, their function is needed only for short period of time. Okay, so basically life determines how long they need to. Why microtubule or tubulin has so much long time? Why actin is so much long time? That because if actin is not there, your structure of the cells will be collapsed. Okay, why microtubule is needed? That is again integrity of the cell structure. You know that basically the major critical role of microtubule is that congregation of our genome during mitosis and equal separation. Okay, so that's why those proteins have long life, like more than 24 hours. Okay, so exactly, so that understanding of the protein is, each and every protein is very, very important. And you know, out of 34,000 proteins, we know in details maybe 50% of them, and 50% of the rest of the 50% we don't know that much. So understanding of the proteins is for understanding the normal homeostasis as well as the disease pathogenesis. Okay. Okay, so this is, I'm telling you, this almost 100 different types of post-translation modification happens. I am telling you only few which commonly happen is that, like lipidation, sumylation, phosphorylation, acetylation, methylation, glycosylation, disulfide bond, as I said, only cysteine. If the protein, two proteins, they will have that from the bigger complex through covalent bond, then disulfide bond should happen. Then hydroxylation and ubiquitination. Already I showed you that ubiquitination for death. But I will tell you that this is not only for death. This is also needed many. And among these, this is true one, that ubiquitination and sumylation, this is actually post-translation modification, but it is the covalent bond between two proteins. One is very small protein like ubiquitin is the 76 amino acid, and sumylation is almost similar. This is, but this is called the post-translation modification. And there actually modification happens in different way. I'll talk about later slide. So I'll talk about the phosphorylation. You know that phosphorylation is very, very important. Okay, for most of the protein, the phosphorylation invariably happens. And there are two kinds of enzymes, or you can say proteins, which are involved in this process. One is called kinase, and one is called phosphatase. And the kinase, we have almost 3,000 kinases and 200 phosphatases. Okay, so kinases, and you know that particularly cancer, the most of the drug developed against kinase. Because if they are not regulated properly, they will phosphorylate their protein substrate differentially. And you know that if there is precise phosphorylation will not happen, more phosphorylation or less phosphorylation will cause the disease. For example, that you know that lung cancer. Lung cancer, there is called EGFR, the receptor, epidermal growth factor receptor, which is highly activated, and phosphorylation happens, and that will phosphorylate many proteins. Okay, and leading to that, the kinase activation pathway, MAP kinase, GRK and PI3 kinase pathway, that will activate unnecessarily, and that will cause the disease called cancer, particularly EGFR. You know, the lung cancer, most of the 70% of the lung cancer cases, the EGFR is highly activated. The mutation happens. Same thing melanoma. Like BRAF mutation happens and its kinase activity is almost 100 times more, and so 80% melanoma cases that kinase, BRAF kinase is highly activated. So they will phosphorylate. Okay. So and in the basically phosphorylation where it happens, in the 3 amino acids where the phosphorylation happens. One is serine, one is threonine, and one is tyrosine. And these kinases, they are actually belongs to 2 families. One is serine threonine kinase, one is tyrosine kinase. Okay. So phosphatase, the kinase, that 3000 and we have 200 phosphatase only. So even your kinase is functioning properly, but phosphatase is not working properly. So again disease will happen, and it is always evident in that cancer. For example, breast cancer, ovarian cancer, pancreatic cancer. Okay, that P10 phosphatase, one of the common phosphatase which is inactivated in the cancer is the P10, and because of that PI3 kinase is highly activated in the breast cancer. Basically 70% kinases 70% of the PI3 kinase pathway is activated because of P10 mutation, P10 inactivation. So it is not only kinase, the phosphatases are also equally important. Okay. Okay. So that one of the example, the kinase or phosphorylation important is the transcription process. As I said, that whatever genetic information stored it, they will be transmitted in the form of the RNA or decoded in the form of RNA. And this process is very, very essential and routine work for the cells to routine to work. So the transcription, what happened? This is the, for example, there is the 3 steps happen in the transcription process: transcription initiation, transcription elongation, transcription termination. So whether it is the prokaryotic, mean bacteria, or others, or eukaryotic, this process will be continuing. So in this process, the phosphorylation in the RNA polymerase happens. Without phosphorylation, this transcription initiation will not start. Okay, even termination will not happen. So here that 2, basically it is the C-terminal domain, repeats domain happen. Here the serine 2 and serine 5. That until unless serine 5 phosphorylation will happen, then transcription initiation will not happen. Okay, even the end of the elongation step, if the serine 2 phosphorylation will not happen, termination will not happen. If termination is not proper, the protein will not form properly. So then again deregulation will happen. So I'm trying to tell you that how phosphorylation, post-structure modification, is playing critical role for the life of a protein. Okay. Similar example, cell cycle. You know the cancer is basically the uncontrolled cell proliferation. Okay, as I said. So here is the cyclic process happens: G1 phase, S phase, G2 phase, and M phase of the cell cycle. In each phase, there are set of kinase. Okay, they will be activated and then inactivated. Like in the G1 phase, the CDK4, CDK6, they will form the cyclin, protein called cyclin D. So they will form the complex, and this complex will phosphorylate. One of the most important protein is the retinoblastoma protein. So retinoblastoma protein is a tumor suppressor. So it functions as a checkpoint if the cells are not properly progressed in the G1 phase. So in this phase, retinoblastoma protein will stop here, not the cell to go forward. And if cell go forward with overcoming this, then there will be a cancer. Okay, so retinoblastoma protein, you know this protein is highly inactivated. So here basically cyclin D, CDK4 or CDK6 complex phosphorylate retinoblastoma protein, then inactivate it, and as a inactivation, then this is the protein is called E2F family proteins. So this will be activated because this interaction will be lost because of the phosphorylation of retinoblastoma protein. Then this protein will actually transcriptionally upregulate many genes which are involved in the S phase of the cell cycle. So if this retinoblastoma protein is inactivated by this protein or other mutation, then this checkpoint will not be there. Okay, so that checkpoint, if it is not functional, then means abnormal cell proliferation will happen. Okay. This is kinase. You see that this is kinase signaling pathway where basically phosphorylation happens. This is PI3 AKT pathway. So you know that, as I said, that AKT is highly activated in 70% breast cancer cases. So if this AKT actually phosphorylated so many proteins like FOXO3, FOXO1A, then GSK3 beta, TCS2. They phosphorylate, basically AKT phosphorylated them, inactivate them to promote the survival, proliferation, metabolism, growth and others for the cancer cells. So that's the reason that this protein is highly activated to actually remove this all the checkpoint. Okay. Then another important thing is that DNA damage. You know that most of the chemotherapy drugs used in multiple cancers, say from breast cancer to ovarian cancer to lung cancer, any cancer you can think about it, most of the primary drugs, chemotherapy drugs are DNA damaging inducing drug. Okay, like cisplatin, many of you have heard. Cisplatin, doxorubicin, etoposide, these are the first line chemotherapeutic drugs. So these drugs, what they do is that in the cancer cell, the genome, they break it so that protein expression will not happen. If protein expression will not happen, cells will die because the protein is the most important worker in the cells. So if they don't are not there, then work will not happen. So that's the strategy generally we use for the treatment. So this is the two. These are the two kinases, the ATM and ATR. So what they do, they actually phosphorylated more than 600 protein, just induced DNA damage within 15 minutes. These two kinases will be activated. Okay, and then they will phosphorylate more than 600 proteins. So think about that, how phosphorylation, so many proteins are getting phosphorylated, and they are, after phosphorylation, actually these proteins play critical role in the normal cells. They will actually, if too much damage happen, cells will be dead, which we need for the cancer cells. But in the cancer, particularly for normal cells, we need cell cycle arrest because normal cells should not be die. Okay, so the cell cycle will happen, DNA repair happen. Each class of proteins are getting phosphorylated by these two kinases primarily. Okay, so we need cell death for the cancer cells, but we need the cell cycle arrest and DNA repair for the normal cells. Why cytotoxic effect comes for the all chemotherapy drugs? Because what happened, they induce the death, at the same time they also affect the normal cells. If normal cells are not prepared properly, that death will be there. There is the cytotoxic effects happen. Okay. So I'll come that one of the protein, I'll come later, that one of the protein called histone H2AX, okay, H2AX, that actually first protein, one of the first protein which will be phosphorylated, and that will help in the DNA damage repair process. I'll come later on the slide. Okay, so how to, since Professor Bhargav told me that there will be the student from BSc and other, so I made it very simple so that you can understand, you can utilize that. Yes.
Can I ask a question?
Sure, anytime.
Sir, can you little elaborate on either death signal or death protein, how they functions?
Okay, so I did not make that slide. Okay, I'll tell you.
Okay, thank you, sir.
So basically, I'll go back. Okay, so what happen, this is ATM, ATR will be activated, and following there are called protein checkpoint 1, checkpoint 2, okay, and DNA PKCS. So these kinases will be activated. One of the protein you know that involved in the apoptosis is P53, which is guardian of the genome. Okay, so P53, when it is basically activated by ATM phosphorylate directly, ATM ATR phosphorylate P53 at serine 15 or serine 20 position. Once it is phosphorylated, it's accumulate, because what is happening? It generally under normal condition, P53 will be hijacked by another protein called MDM2. Okay, hijacked happen, it will not allow the P53 to function as a apoptosis inducer. Okay, so but when the drug like cisplatin, doxorubicin, etoposide, like that kind of drug, 5-fluorouracil, all cancer drug if you give, then what will happen? This DNA damage will happen, that will activate ATM. ATM will phosphorylate P53 at serine and 15 and serine 20 position, and once it is phosphorylated those two positions, then MDM2 cannot hijack this one. That will be removed from P53, removed from MDM2. Then P53 transcriptionally activate PUMA. That is called another protein called PUMA, P53 upregulated protein. So that PUMA actually form complex with another protein called BCL2, BCL lymphoma-like protein 2. So BCL2, once it interact, it will release the another protein called BAX beam. And BAX and beam, they form the oligomer within the membrane of mitochondria, outside the outer membrane of the mitochondria. Once they oligomerize, they form the pore in the mitochondrial membrane. That membrane potential will change. And then from that, once membrane potential is changed, you know that mitochondria is double membrane, inner membrane, outer membrane. So outer membrane pore will happen, but inner membrane and outer membrane there is a APAF protein, okay, and cytochrome C. That will be released, and then caspase activation will happen. Caspase activation will happen, and then what will happen? Once caspase activated, so DNA will be also cleaved, and apoptosis will happen. Cells will. This is the entire signal. PUMA will be phosphorylated, PUMA will be acetylated. All these post-transition modification will happen. So, but trigger will happen to the P53. First initial phosphorylation in P53 at serine 15 and serine 20 position is very, very critical to induce the apoptotic cell death. There are other way of cell death is there, like autophagy, necrosis. So these things happens. That will happen. There is a different mechanism altogether. There is a ULK kinase will be there. That will phosphorylate a backlin. Backlin will form the membrane initiation for autophagy system, and then autophagosome and lysosome will be actually merged together, autolysosome happen, and then engulf happen, then the autophagic death will happen. So necrosis also similar way. There is the oxidative stress should be there, and then membrane will be ruptured, and then death will. All process are basically triggered by these different types of post-translation modification. Okay. Yes.
What would be the stimulus that triggers either apoptosis or
Apoptosis, for example, two kinds of apoptosis happens. Intrinsic pathway of apoptosis or extrinsic pathway of apoptosis. Your cells, basically the ligand based, extrinsic, fat ligand. Okay. So if ligand is getting highly expressed, that extrinsic pathway apoptosis happens. Intrinsic pathway, there are many things. One is that oxidative stress, reactive oxygen, because of metabolic defects happen within the cells, then reactive oxygen species will induce. Based on the reactive oxygen species, if it is too much superoxides there, then cells will go for, basically that is the trigger. That is the trigger where basically too much oxidative stress happens, cells will go for necrosis. If it is oxidative stress are moderate, cells will go for that apoptotic cell death. If it is cells are nutrient deficient, metabolically nutrient deficiency, cells will go for autophagy. So these are the stimulus. Dependent stimulus. If the DNA damage happens, cells generally go for apoptotic cell death or senescence mediated cell death. Okay. So depending on that cells, what kind of insults cells experience. Okay. This is the cell's intrinsic sense that it is too much toxic. Okay. What we know, if it is too much acidity, we know generally 15. Okay. If it is not that much with digen or something. So like that, cells senses what kind of stress. Too much oxidative stress means basically disintegrated. Too much damage. Cells cannot repair so much damage. Okay. So cells give the signal, go for death. And if ask the cells to what death you want, cells will prefer apoptosis rather than necrosis because two reasons. Necrosis, what is happen? Entire cells will be damaged, all membrane will be ruptured, everything will be go to the surroundings. And if your waste material is going all the surroundings, what will happen? It will, disaster will happen. So the inflammation started. Whereas apoptotic cells, nothing will be, cellular things will be out. So it will be in the, within the cells. Then macrophage and others, phagocytic cells, they will capture, they will engulf, and that so that our surroundings should not be affected. Senescence will happen. What is happen? Mild damage happen in the cells. Cells thinks that it can be repaired. Let us do. Okay, like you see that all of you have that ventilator in the hospital. You know that patients are inhaling. Ventilator, what is happening? There is a chance of recovery 4%, 5%. Okay. So that stage called the senescence, there is a chance of the coming back. And 90% senescent cell will be die. And few percentage may come back as happened in the ventilator. Okay. So basically mild DNA damage happens. The cells will try to the DNA damage repair machinery to come into, fix it. If fix it, that time cells will be arrested at the G1 phase of the cell cycle called the ventilation. Okay. And then arrest happen, give the longer period of time that repair happen. If cells think still it is not that, many times you have, okay, go ahead. Because many of you have encountered that people are ventilated for month after month after month. Okay. And then no chance of recovering. Okay. So that is the death ultimate, this one happened. This is the cue, depends on the cells how it is experiencing. Okay. Thank you, madam. So how to detect that? Now I am coming to the research oriented things, that how to detect your phosphorylation. So many phosphorylation happens. So, oh sorry, whatever. So three different techniques are available to detect that phosphorylation: immunofluorescence, immunoblotting, or mass spectrometry. Okay. So immunofluorescence is that when you can do, if you have, first of all, you have to have microscope. Without microscope, immunofluorescence cannot be done. Second requirement what we need is that your specific phosphospecific antibody. Okay, phosphospecific antibody is there, then and then you can detect by that immunofluorescence. Basically, for example, target protein means, suppose for example P53. If you have antibody anti-P53 with serine 15 or serine 20, we probe with them, then you secondary antibody will be conjugated with the fluorescence marker, and then you under the microscope you see. So good thing about it, the specific whether basically whether it is a cytoplasm, nucleus, where it is, you can find it out. But disadvantage of this technique is you need to have specific antibody and microscope. If you don't have, you cannot use this technique. Then Western blotting. This is a simple technique again. Okay. So what happened? You take that basically cells, lyse them, boiled them to denature the protein in the presence of SDS. Why you add SDS? SDS actually two things it does. One, denature the protein. Denature means, if you see, this is the my structure, native structure. If you give, they will be unfolded like this. Denature will happen. Second, what will happen? Because of this SDS, all proteins will be negatively charged, uniform negatively charged. Okay, so that when you put in that in the electrophoresis, based on the molecular weight, they will be separated because all are negative charge. So they will transfer from negative positive, direction will go, and based on their molecular weight, okay, they will be separated like this. And then you transfer in the PBDF membrane and then probe with the primary and secondary antibody, and then you see. So exact how much proteins are getting phosphorylated, you can measure quantitatively. Okay, so that is the major advantage of this one. And if you do the lysis in the fractionation way, in the mitochondrial function, membrane function, nuclear function, cytoplasmic function, any type of fractionation you do, so that kind also you can see that which organelle, where it is getting expressed. Okay. So this is a less costly instrument you need, okay, compared to the immunofluorescence microscopy. Okay. So this is a versatile technique, okay, you can use. But again, disadvantage is that you need to have the specific antibody against your protein. Okay. So now question is that, can we detect a phosphorylation if we don't have specific antibody? Okay. Can you do? Because 30,000 antibody, specifically the phospho antibody, modified antibody is not available. But we have to do research. We can do many ways. Two ways I can show you that. One is mass spectrometry. This is the versatile technique again, but costly. Okay. So because one microscope minimum 4 crores you need to have it. Okay. So here again cells, protein extract as we described previously. Then there is a phosphoprotein enrichment. There is called titanium oxide bead. So you enrich only phosphoprotein. Okay. And then you digest those protein with the trypsin and subject to the mass spectrometry. So you will get that what are the proteins are getting phosphorylated. And in the mass spectrometry, which serine, which threonine, which tyrosine is getting phosphorylated, that also you can detect from this. Okay. So that is the advantage of this, the specific residue. Okay. Without antibody you can detect, but as I said, it is a costly one. Okay. You can do that even gel based also you can. Okay. So when can we do that? Suppose we don't have mass spectrometry, we don't know is that protein, but we know the protein antibody which we have, but we don't have phosphospecific antibody. In that case also, you do the protein extract, pull down specific protein like P53 by immunoprecipitation, and then immunoblot it, and there is called pan-serine, phosphoserine, pan-phosphothreonine, pan-tyrosine also. You see, your protein is getting phosphorylated at the tyrosine residue, threonine residue, or serine residue. Once you know that, okay, this is my giving the serine phosphorylation level. So now in the protein, you see the how many serines are there. You mutate it. By molecular biology technique, you mutate it. And see that if you mutation, you don't see the signal, that means you can identify like that way. And sir is here, that Japan, one paper, you will see the 60, 70 mutant protein they generally make. Simple that. Say, the wonderful, their student are, I don't know how much they work, basically skip one protein, they don't care about making 70, 80 clones. Okay. So that is emerging. Here, one student you ask for more than 5, no sir, too much work. But Japanese student, I don't know, they are wonderful actually. Okay. So then next is the acetylation. This is also widely happen in the protein. And I'll show you that how acetylation is very, very important. So acetylation is nothing but is the acetyl group addition to the protein group. So lysine NH2 group basically will covalently link with the acetyl group with the help of acetylase. And this acetyl group is provided by acetyl-CoA. Okay, coenzyme. So this addition happen, and there is a deacetylase which remove acetyl group to make it free lysine. So always basically like our the cellular system, there is a positive regulator, there is an always negative regulator to make check and balance. Okay, so this happen. Now I'm coming to the acetylation. You see the major happens is the depending on the resolution, that if it is a single chromosome, is the 1400 nanometer resolution. If you find resolution to 2 nanometer, you see the double-strand helix. And here, nucleosome is there. Nucleosome is the octamer of histones. So there, histone H2A, 2B, histone 3, and histone 4. So all of them has a tail, okay? And these are basically, I'll skip there, you don't need heterochromatic. So all of them have that tail. Okay, this tail have several lysines. Okay, and the lysines are basically susceptible to multiple type of post-translation modification: acetylation, methylation, hydroxylation, ubiquitination. And these tails, based on that, the post-translation modification status, they will be either heterochromatin or euchromatin. So basically the black spot you are talking about all are heterochromatin, and then blue, this curve is the euchromatin. So euchromatin means active genes. They will transcribe, their transcription will happen and protein synthesis happen. But heterochromatin, their transcription will not happen, protein synthesis will not happen. So heterochromatin and euchromatin depends on histone tails post-translation modification like methylation, acetylation, ubiquitination. Okay, these are the 3 different post-translation modification I'll tell you. So these are the basically lysine 9, lysine 14, lysine 18, lysine 23, histone 3. They are getting acetylation. Same thing histone 4. So these histones basically acetylation or methylation, they can form part of the euchromatin or heterochromatin depending on the position. Okay, the position maintains the euchromatin or heterochromatin. So here is this, that you see the acetylation, this one is the K4 or K14. So basically that is the euchromatin mark. Okay, it's that H4, these are the cases is the hetero. That acetylation is this is the H2A, lysine 5, that acetylation is the heterochromatin mark. Means position as well as that type of position modification that will tell one gene will be active or inactive. Active or inactive in the sense is transcription will happen or not happen. If transcription will happen, means genes are active. If transcription will not happen, genes are inactive, means heterochromatin. Okay, so now coming to the glycosylation. This is one of the most important post-translation modification if you think about our immune system, cytokines. any secretory protein which need to be secreted, means out of the cell. Okay, so that need to be glycosylated. Without glycosylation, not a single protein will be extracellular. Okay, so all cytokines, majority of the cytokines will be glycosylated. Membrane protein, at least 50% of the membrane protein will be glycosylated. Without glycosylation, their membrane will not be, they will not be recruited to the membrane. So this is one of the important post-translational modification, because T cells, cytokine secretion, so that will happen only and only glycosylation happens in the endoplasmic reticulum and then Golgi. If it is Golgi, their maturation will not happen, they will be degraded by proteasome or lysosome. Okay, so that's why the glycosylation is one of the very important post-translational modifications. So I'm saying that two types of glycosylation happen. L-linked glycosylation, this is the, oh sorry. Bhargav, can you make it? Yes. Yes. Okay. See, N-linked, the asparagine, N-linked glycosylation happen. Okay, and O-linked glycosylation, serine and threonine will happen. These two types of glycosylation happens, but there is no antibody. We see there are antibody for acetylation, methylation, phosphorylation, ubiquitination, but glycosylation specific antibodies are not there. So their detection system is little bit of difficult, or you see that migration in the western blot, their migration change. If it is the glycosylation, generally 10 to 20 to 30 kilodalton protein size will increase. Okay. So detection of glycosylation, most of the cases acetylation or immunoblot, basically migration. Okay. So one of the glycosylation things I'll tell you, which is currently in the highlights in the clinic, is that cancer, basically anti-PD-1 therapy or anti-PD-L1 therapy. This is the immunotherapy which is used in the clinic, is there particularly for lung cancer and melanoma cancer. There are almost 25% patients cured because of these two targets, the PD-L1 and PD-1. And they are highly glycosylated and they recruit in the membrane. In the PD-1, that will be in the T cells, cytotoxic T cells, and PD-L1, which will be the surface of the cancer cells. So they form the complex, make the cytotoxic T cells, means T cells which is kill the cells, abnormal cells, that is cytotoxic T cells. So that they will and make the T cells non-functional. Okay. Is the anti-PD-1, so it will bind to the PD-1 basically receptor and then prevent this PD-1 PD-L1 interaction, so that cytotoxic T cells will be active, then it promotes the cancer cell to die. Okay, so this is the antibody therapy. This is that, see why nature has designed this, that nature has designed this axis to prevent the autoimmunity, autoreactive, hyperreactive. To prevent that nature has designed this axis. But cancer cells, I always say one bad term, cancer cells is equal to terrorist. Okay, so they always hijack or always kill who are against them. The T cells are against them. Okay, they take the revenge, make the T cell to die. Okay. So that is the case, so glycosylation, and I'll show you that one important thing. So the PD-L1, okay, so PD-L1, you see when in the glycosylated form, its molecular weight, this is the non-glycosylated form, okay. When it will be glycosylated, its molecular weight almost 20 kilodalton increase, because as I said, there is no specific antibody developed against the glycosylation. Okay, only you have to see that whether it is glycosylated or not through this is migration difference only till that. Then last part I will say that how much time? Okay, okay. Okay, sure, sure, thank you. So this is the last part, that ubiquitination. Ubiquitination nothing but a conjugation of ubiquitin, which is 76 amino acid protein. Okay, so this was first discovered, 1975. Okay, so this is a ubiquitous protein. This is in the Harvard. What simple experiment they did? They took that different organ of a guinea pig, ran to the simple SDS page, and they found the one protein which is common to all the organs of the guinea pig. So this is ubiquitously expressed. So first they have given the ubiquitous. Okay. Later on, basically they developed the structure and they found that there are the 7 lysine residues are there, and this is the 76 amino acid, and based on their functional characterization, so these 3 scientists got the Nobel Prize in 2004. Okay, so this is the wide importance. So he showed that this ubiquitin plays very, very important role in the immune response also. Okay. So if you think about the ubiquitination, it is important for all sorts of things: the signal transduction, immune response, cell cycle regulation, DNA repair, DNA damage response, like all the metabolism, everything it controls if it is getting ligated to the other protein. Okay. As I said, cell cycle, why if it is so important? If you that, because each cyclines, they will be very short period of time. If the longer period of time they are there, then cell cycle will be defective. I will show you some example in our lab. Okay. So this is the ubiquitin, the primary structure. Okay. So any protein can be monoubiquitinated, multi-monoubiquitinated, or polyubiquitinated. Poly means minimum 4 ubiquitin conjugation will be there, and this number 48, 11, K63, like that, K29. So as I said, these are number is actually numbering here, the K6, K11, 27. When ubiquitin conjugating through lysine 29, that will mark as a lysine, means polyubiquitination through lysine 29. Okay. So there will be linear branching there, but we are not till date. We know only functions of these and these two. We don't know the function, what is the importance of this linkage, or this linkage, and there is no antibody. There is no antibody for these linkage ubiquitination. Only antibody is available for this and this. So what they do? What is the importance now? We know. See, basically ubiquitination happens in the 3-step process. One is the activation step. The ubiquitin will be activated in the presence of ATP by E1 activating enzyme, and we have only 2 E1 activating enzyme in each cell. And then E2 will be that this activated will transfer to E2 conjugative enzyme. We have almost approximately 50 E2 in our cells. And then this activated ubiquitin will transfer to the target protein like P53 or any other protein with the help of E3. And we have almost 600 E3. They are different class. Okay. So I will not go through details. That this is one is the SCF complex. So this ubiquitin protein, okay, will transfer to that 26S proteasome further process. With the cell, this protein should be die or it will be alive, depending on the linkage. If it is the lysine 48 or lysine K11, cells will be die by degradation. Okay, in this process. If it is a lysine 63, it will be signal transaction. It will not be degraded through this. So here again detection is the immunoblotting and mass spectrometry. If the polyubiquitination happens, you will get the ladder. If a monoubiquitination happens, you will get single 7 kilodalton shift. If it is 2, it is 14. If it is 3, it will be around 21 kilodalton shift. And if it is more than that, that means it is getting polyubiquitinated. Okay. So cell cycle, as I said, the cell cycle, the ubiquitination plays very, very critical role. Like cyclin D, at the end of the G1 phase of the cell cycle, cyclin D need to be degraded. So that time it will not, it should be polyubiquitinated. Similarly, cyclin A at the end of that G2 phase, prophase, the cyclin A need to be degraded. At the initiation of G1 phase, cyclin E need to be degraded. So that will be polyubiquitinated, and then degradation will happen. So this is that basically cell cycle fact sorting you can do. And as I said, if here I am showing the cyclin A, which need not be there in the G1 phase of the cell cycle, but if you disrupt that E3 ligase, which basically target the cyclin A for the degradation in the G1 phase, so then cyclin A will be upregulated, and that will be premature replication initiation will start, and that will make the aneuploidy, that is the aneuploidy happen. So that means precise cell cycle regulation, each and every proteins need to be precisely activated, means their synthesis will happen and degradation will happen. If they are premature synthesis will happen, that kind of disaster will happen. Okay. So signaling, you know that colon cancer, Wnt signaling is much more, and that is because of KRAS activation. So this one, in the normal cell cycle, basically beta-catenin will be in here in the membrane, and that will be degraded, that will prevent to the nucleus, go to the nucleus. So and that actually beta TRCP1 actually target promote the polyubiquitination phosphorylation followed by polyubiquitination, it will be degraded, so prevent that nuclear translocation. In the cancer, what will happen, the Wnt activated things will happen, this will be beta catenin will be inactivated, and then that beta will be activated by inactivation of GSK3 beta, then beta catenin will translocate to the nucleus, and that then it will be activated and it promotes the cancer. Okay. So that ubiquitination is very important in this to prevent the overactivation of Wnt signaling pathway. Okay. So then another ubiquitination I am talking about the transcription process. As I showed, that entire whichever the active genes means euchromatin, the entire things that promoter to termination, the H2B will be ubiquitinated. If it is not ubiquitinated, that genes will not be transcribed. Okay. So cell cycle arrest DNA repair. So I am going through that ABXO31. So we showed that it is actually degrade that cyclin D1 into cell cycle arrest. Then whether it has a repair activity or not, we did not know. So we found that basically if I deplete that ABXO31, that you know that it is the gamma H2X. These each foci means there is a DNA damage happened. DNA damage happened, and that should be repaired. So here this signal will happen and DNA damage repair machinery will be recruited, and with time the repair will happen. But if FBXO31 is not there, see damage foci will be significantly reduced. So that means repair will not happen and cells will die much faster. So then we saw that if FBXO31 is not there, then recruitment of repair protein Q80 will be improperly happens. And then this is another protein called the RAD51. That recruitment compared to this, the recruitment is significantly reduced. So again, repair will be ineffective, and so cells will die. So this is, I am showing that here this gamma H2X getting ubiquitinated by probing. The each position, the foci, those are ubiquitinated. So ubiquitination is important. If we deplete the FBXO31, ubiquitination is not happening. That's why the foci is not that much happening. So repair will not happen. Okay. So basically what I am trying to tell you is that all these post modification, each post modification is unique, and they play critical role in that cellular, and they determine the cellular functions of protein, and they play critical role in the cellular processes. So many processes I have discussed. So this is my lab. Okay, actually, few of them. These two people moved to Harvard, and he has also he has gone to the NIH. So rest of the people are working. Oh no, he has she has gone to that New York, New York. So newcomers are not there. This is 2020 picture. So these are my collaborators, and then I'm having the fund from that Department of Biotechnology, Department of Science and Technology, CSR, and ICMR. Okay. Thank you, and I'll be happy that you question. Sure, sure, sure.
Previously, we used to study the endo-reduplication process in plant. So at that time, we have observed the role of plant hormones, particularly auxin has a direct relationship with activation as well as suppression of the cyclin D kinase analog in a plant.
Okay.
And the same pathway was also regulated and controlled by other hormones, particularly gibberellin and cytokine.
Okay.
So, is there any same mechanism or analogous mechanism exist in animal cells or human cells, for, particularly through hormones?
Hormone definitely, but that role of hormone in the cell division not that much explored. Not that much explored. This is basically growth factor. Means here animal cells that basically division is controlled by growth factor.
So
Okay.
what we have observed that when we provide the cytokine, particularly arrested cell, when we provide the additional cytokines externally, the process is reversed. The
Okay.
endo-reduplication process get reversed.
Okay. No, in the one cytokine I can talk about is the TNF alpha.
Yeah.
But here what is happening, TNF alpha not cell cycle, it actually activate indirectly the cell cycle through NF-kappa B pathway. NF-kappa B pathway, the cytokine I know that actually activate TNF alpha, and TNF alpha activate NF-kappa B signaling pathway. Depending on the context, TNF alpha can promote or it can inhibit. This is the context dependent, which context it is happening. Okay, so that one I know, but other I don't know really.
I will discuss it.
Sure, sure, sure.
Thank you, sir, for an enriching speech about all the life cycle, the entire life cycle of the proteins. And we feel truly, deeply fortunate to have a person of your stature to inculcate knowledge to us. Now I'd like to request Dr. Rohan Pandya, sir, to kindly facilitate Dr. Manas Kumar Santra with a memento and a certificate. Thank you, sirs. Now let us quickly move forward with our second expert talk of Dr. Harish Kumar Madhyastha. Dr. Madhyastha is a faculty professor at Department of Cardiovascular Physiology at the University of Miyazaki, Japan. He holds 2 PhD degrees and he pursued his postdoctoral career at MCRC Chennai. And after completing his postdoc at Miyazaki University, it culminated as a faculty position over there. He has presented conferences and proceedings for more than 100 times, and he has picked up frequently national and international awards and media. He is an awardee of Fellow Biotechnology Research Society India. He is Fellow of Royal Biological Society London and Bharat Shiksha Gaurav Puraskar. I would cordially invite sir for continue with his expert talk. Thank you.
Thank you very much for the organizers and the management of Atma University for this August House, which is been conducted very quickly by two young coordinators of Atma University. Congratulations to you and my dear faculty friends and adorable students. Thank you, Professor Manas. You reduced my task of explaining the cell, what is inside the cell, so that I'll quickly go to the what is happening if a cell is treated by nanoparticles. नहीं पॉइंटर में नहीं चलते वैसे नहीं रहने दो ऐसा रहने दो Yeah, this is my academic yatra. No, go back. So I born in Karnataka, a coastal village, and moved to Maharashtra, Pune, for Amrit Biotechnology as a scientist. Then came to your own place, Rajkot, for master's degree. Then I moved back to MCRC. It is a Pari Agro Industries R&D unit, backside to the IIT Madras. Then I went to Bangkok, AIT, Asian Institute of Technology for the postdocs. Then directly came to the Miyazaki. This is my Karma Bhumi. So Janma Bhumi to Karma Bhumi. Next. Where is Miyazaki? Nobody knows, I think. You must have Googled yesterday after seeing the pamphlet. It is a southeast portion of the nation. It is in the Japan, Hokkaido, Honshu, Shikoku, and Kyushu. This is the smallest one. Nagasaki is here, Miyazaki is here, Tokyo is here. Yeah. Next. A beautiful countryside. Rice bowl of Japan and radish bowl of Japan also. Beautiful countryside. See rice field. And excellent Pacific Rim. Good for honeymoon. Right? And we have not only that spot, excellent spot, and we have the ocean immersed temple. It is a cave temple and excellent hill-locked paddy fields and living system, social engineering, with 5G internet in all villages. Next. Our university is Miyazaki University, started way back at 49. It is a national importance university, institute of eminence. That's what you call in Japan and in India. But renamed as the Miyazaki University Corporation because they have touched the entire education system has been changed in 2003. All university, although it is a government, we should work like the corporate system. So national universities, we have 2 campuses, graduate schools, 5,500 local student strength. Out of which 680 teaching staffs, teacher-student ratio is 1:8, 8.1. It is almost equal to the global standard. Yeah. International outlook, we have 112 academic exchange agreement between all over the globe, especially the 32 countries. Atma University is one among them. So we inked MOU in 2017, just before Corona. Go there. Next. This is my Google Scholar. Next. Directly I will hit to the subject now. What is nanoparticle? Nano, because cell, professor explained, is the cell. What is that? You students may be knowing what is the size of the cells. It is in the micrometer. But this is the nano, one step further, 1/1000, still down. It is the nanometer. So nanoparticles are explained as any particles which is highly invisible ordinary in the ordinary microscope also, but in electron microscope you can view the particles up to 100 nanometer. Any particles which is up to 100 nanometer is called the nanoparticles. Classification is a one-dimensional, so where x to x and y to y is in the nano size, but L is not in the nano. It may be a meter or kilometer, whatever it is. The classical example is nanowire. Nowadays, the concept is originated in the world that you install the solar power in the moon, through the nanowire and electroconductivity phenomenon, you can transfer that energy into the earth. Research is going on in the Harvard and Boston, Stanford, and in Tokyo University also. Next. Two-dimensionals where L is in the Lx, length x is the nano size, Ly is not, but thickness is in the nano size. The classic example is your iPhone thin layer cover. It is a classic example. Next is the global one. It is a crystal globe where the globe itself is not nano, but each particle, each embedded or linked particle inside the globe is the nanoparticles. If you break this globe, it is equivalent to the 1 football ground area. Next. Where is nano? Nano is universal. You can see it in chemistry, you can see it in bio, you can see it in the space, you can see it in the physics. It is universal. Since I am a biologist, I focus only on these portions. How this one helps in the microbiology, biomedicines, and other areas of the regenerative medicines. Next. This is the properties of nanoparticle. We can have the nanoparticle of the metals. Basically, metal nanoparticles are, I am interested, silver, titanium, selenium, and ferrous, zinc, and other gold and other things. We encapsulate with the liposomes and the miscellaneous. It is the compositions. The size, as I mentioned earlier, it is 1 nanometer to 100 nanometer is the size of the defined size of the nanoparticles. Sometimes we get the publication for my reviews from I don't want to name the nation of that particular nation. They mentions it is a nanoparticle, but if you see clearly their image, because nowadays the great publication, you should submit the original figures also. Otherwise, they will not accept it. If you see their original backup file, it will be more than 100 micrometer. But they say it is nano. Direct rejection, even though he is a Nobel laureate, no problem. The instruction from the publisher is very clear to the editors, associate editors, that kindly check the supplementary document and reject it. That's all. So it should not be more than 100. Some up to 120, 125, 150. Am I right, Professor? I think 150 can be called as nanoparticles, but not more than that. Not micrometer, not accepted at all. And the surface chemistry, where I am working, we can ligand the surfaces of any metal particles with the proteins, with the positive negative charges or negative charge, neutral charged or the zwitteric ionic properties particles to have the combined local effect on the cell. And shape will be the sphericals, cubical, polyhedronals, rod shaped or disc shaped. Can be anything. And any size can be a liganded or a conjugated on the surface of the metal nanoparticles or any other particles. Next. This is the functionalizations, how the central dogma of in my case it is a metal. The metal is the coronized with biocompatible linkers, with biocompatible molecules with the linkers, antigens, and fluorescent signaling and shape recognition molecules. This is useful for the sensors molecules, sensors devices. Next. Why nanoparticle is coronated is required? As I mentioned just now, you have the hard core of the metal and there is a soft protein corona or lipid corona or whatever it is. There's the middle which links the metal particle with the interested protein which has the direct relevance on the specific disease. Each professor, as professor explained, protein has important role and it has a life cycle in the managing the cellular hemostasis mechanisms. If the cellular hemostasis mechanism, as he explained, you have the different regeneration or the degeneration phenomena in the cell. To control that regeneration, the specific one is the ROS, reactive oxygen species mechanism. This is the starting point, Gangotri of the disease. As I mentioned, as I used to tell everybody, to control that ROS, we should have the specific colonization of the interested proteins or some drugs, and we will have the controlled release of the drug with the specific pinpoint tissue through the cellular uptake mechanisms. These are all the effect of the nanoparticle behavior, how it controls the toxicity and targeting efficiency, accumulation in the filtering organs. One more important is the most danger of this nanoparticle is the accumulation in the glomerulus or kidney. The glomerulus filtration mechanism failed, your kidney will be a metal mine tomorrow. That is most dangerous. One should control the accumulation of this nanoparticle in the body. Next. This is the metabolic trajectory. That means how nanoparticles, of course, we are not seeing in our environment, but in environment is the most, we have lot of nanoparticles which are suspended in the molecules. Especially, you must be knowing, you must have heard or learned in the classes, in your credit, in MSc or PG credit, it's the nanoplastics. Nanoplastics are the most important nanoparticles which are in the environment. It's most dangerous to the human being. We will not know what is happening in our lung. Right? The best example is asthma. So, exposure pathway, it is the ocular, from the eye, from the inhalation, from the nose, the oral, drinking, and dermal exposure, the most dangerous, and intravenous injection. And once it entered into the body, you will have the organ or tissue specific interactions. For example, nanoparticles, if you inhaled one, direct effect is on the lung. Then, as Professor mentions, once it goes to the lung, it comes to the blood circulation. If it is an inhalation mechanism or oral inhalation mechanisms, then it interacts with the cell. Nanoparticle interacts with the cells. What happens to the cell? That's what he just explained, beautifully explained, what is the internal architecture of the cell. Each organ or specific organ will be destroyed, except nucleus, I believe. So nucleus, I think, is the most protected, junction in the cell. But nowadays, there is an imbalance in the nuclear pore is also there. There are a lot of work is going on. Nuclear pore imbalance or nuclear pore enlargement have some effect on the entering of this nanoparticle into the nucleus also. Once it goes to the nanoparticles, it may have the delegrative or generative effect on the cell. These are all the examples. Yeah, next. These are all the physical, chemical, and biological effect, how ROS is generated once it goes to the cells. Singlet oxygen, superoxide radicals, oxygen radicals, peroxidized, hydrogen peroxide, and hydroxyl radicals. These are all the radicals of the highly dangerous radicals to ignite the ROS. And it has to be degraded. The nanoparticle has to be, metal ions and organic species has to be degraded in the have to have the specific effect. And as he mentioned, and Professor also asked, some signaling pathways, MAP kinases, NF-kappa B pathways, and PKC kinase pathways, protein kinase C pathways, and SERC-C, and HIF1, NRF2, so many pathways are there. All will have its own role in the maintaining the cellular health. If there is a health is degenerated or health is disturbed, the nanomedicine will come to have the specific effect on this one. There are so many nanoparticles and nanoparticle embedded medicines are in the clinical trial stage, except dermal exposures and beauty exposures. All other nanoparticles are in the clinical stage, but it has not come. In the cancer area, some nanomedicines are available, and cisplatin, especially cisplatin, they converted into the nano-sized with the specific cancer-treated drug. It has come to the market now, but effect still under question mark. Next. So it is a cellular toxicity, just I mentioned, and how it damage the intercellular particles and damage the plasma membrane. Most important one is if a nanoparticle comes into touch with this one cell means, yes, you all know plasma membrane has the lipid layers. The lipid layer, it makes the hole in the cell and particle enters into that one. If that hole is not healed, you have the problem. Lipid peroxidations and ROS generation, these are all difficult managing. It will be very difficult to manage the lipid peroxidation system in the cell. And as I mentioned, this one, damage or failure of the tissue organs, lungs, liver, brain, spleen, kidney, and skin. Everything will be there, which ignites the inflammations. As Professor mentioned, the cellular inflammation will be there. If the inflammation ignited, the first target signal transduction mechanism is NF-kappa B. NF-kappa B will be most dangerous pathways. If it is ignited or if it is overexpressed, you will have multiple diseases. Next. So these are all the nanoparticles, which are different strategies, the actions, how it is interact with the cells. And as I mentioned, ROS, RNS react to nitrogen species, and RSS is the most important junctions which have the different cellular signaling mechanisms. But I am working on the apoptotics, as Professor explained nicely, and necrosis. Necrosis is the damage of the cells, killing of or the death of a cells, which is the inflammatory mediated. And pyroptosis is another mechanism of the cellular death by the inflammatory NLRF mediated cell death. And nowadays, another one apoptotic mechanism has come. It is called ferroptopsis. The ferroptopsis is the only ferrous imbalance in this in the cells. How that ferrous imbalance in the cell will have the degenerative mechanism on the cellular level. And application is the blood cells, dermal cells, skin generations, and cancer apoptotics. Till now, I was working on the skin mechanisms, wound healing, and melanoma, and other things. Cancer, of course, everybody is working. I am also working for the other projects. Yeah. Next. These are, I will not repeat once again the informations. Once nanoparticle enters into the cells, which have the compartmentalized effect on the cellular organelles, mitochondria, nucleus, cytokines, DNA damage, Golgi apparatus, and all other things, and vacuoles also sometimes. Right. Next. Mechanism is by the phagocytosis or the phagocytosis. Here it is the phagocytosis mechanism. For example, if I have opsonine, nanoparticle which is targeted for the opsonine will have the molecular integration of the nanoparticle embedded opsonine. It is called opsonizations, which enters into the by the phagocytic mechanism by the opsonine receptors molecules. Then it is a ligand-based interaction is also there if the nanoparticle is coronized. It is called the phagocytosis, right? Next one, phagocytosis. It is a cell drinking and cell eating. In simple terms, one is cell drinking and one is cell eating. So it is a phagocytic mechanisms, how nanoparticle enters into the cell, inside the cell and have the effect on the cellular mechanism which is targeted for the specific disease. Next. Simple health strategy of this one. Healthy cells, tier 1 is the NR-alpha to the phase 2 enzymes. We'll have the defense mechanism. Tier 3 is the MAP kinases. Finally, it is the all problems with the gene and other things. Next. Omics in the nanoparticle is, these are all the omics. Many people are working, metabolomics, proteomics, lipidomics, and transcriptomes. You can select the one, each one, how the nanoparticle changes the genotypes into the phenotypes. Professor explained very nicely that how protein have the translational, transcriptional, mRNA transcriptions, translational and protein and tissue. The strategy of the proteins. And the same thing we can do. We can start with the genotypes up to the phenotypes. To identify the mechanism of the nanoparticles effect in the gene level, first to identify it. If there is a changes, then dig out the information till the phenotypes. Next. Metabolomic activities. As he told, mass spectrometry is the versatile tool to understand anything inside the cell. Versatile tool. Only thing is it is very costly, but it is very good instruments. Next. Yeah, wound healing. This is my subject. Wound, as you know, cut or a damage in this one. Reverse is healing. Simple mechanism in simple terms. This is the pictorial. Next. Degree is there are clean wound, contaminated wound with good health, and dirty wound. It is a diabetic wound. Next. Fibroblasts are the important master molecules of the wound healing. For the cellular, in the wound healing, only fibroblasts and keratinocytes are important cells for the wound healing. This is how all the interactions or the bicommunication pathways between or communicative pathways or cooperativeness pathways between the keratinocytes and fibroblasts. This we have published in the PLOS One. Next. So I will concentrate only on two cells now. Keratinocytes. How keratinocytes communicate with the fibroblast with the help of TGF-beta and IL-1. And how this one next, how the epithelialization is the initiation of the wound heal and finally heals the wound. Next. If a diabetic wound or a diabetic patient is there, it increases the TGF-beta, very simple. And if the increases in the fibroblast, there is the discommunication or miscommunication between the keratinocytes and fibroblast through this mechanism, TGF-beta and IL-1. If it is there, miscommunication happens between the keratinocytes and fibroblast, which will have the delegative or bad effect on the epithelializations. That means initiation of the cellular repair of the keratinocytes or fibroblast will not be taken place if there is a problem in the epithelializations which will not heal. The classic example is diabetic. Next. My hypothesis is the diabetic retards the fibroblast cell migration is my hypothesis. That means what happens to the cell migration phenomena when there is a diabetic conditions of the disease through the mechanisms of the cellular mechanism or cellular communication mechanism between the keratinocytes and fibroblasts. I will concentrate only on one topic. It is called cell migration. Next. This is the classic video on live cell migrations on it. Yeah. Not possible at all. Click it. Yeah. Click it. No, it's something. Sir, it's problem with the Mac and other things, right? Anyway.
Sir, may I ask one question related
Right.
with the same?
Who
Sir,
is asking?
here.
Yeah.
Shivani ma'am, one last slide, please. Sir, last one, please. That one. Sir, if any person who is taking insulin currently now to cure that diabetic, so at that time the TGF beta level remains as it is, as like the normal one, or it increase or decrease something? Because the diabetic issue is with respect to the insulin and the mechanism is going on. I am currently is diabetic and I am having an insulin, external insulin with me. So is there any effect with respect to this or not? Means it will...
Technically, there should have the positive effect. That means it should increase because you are reversing the cellular damage to the normal this one through the insulin mechanisms. I think it is there, but I did not read that one. I also don't know the clinical aspect of that one, and it is not my research also. So technically, hypothetically, yes, it should have the positive effect on the TGF beta and IL-1 pathways, right? Yeah.
Hello. Sir, as you mentioned ROS, so if ROS infects our body and causes inflammation, does our body have any anti-fighting mechanism, like to fight against the inflammation and all,
Yeah.
or we have to provide external nano medicines?
No, there are cellular antioxidant mechanisms are there. Through that mechanisms, it will try to improve the oxygen hemostasis in the inside the cell. Antioxidant, superoxide dismutase, Professor mentioned. Yeah, there are many antioxidant mechanisms are there. It will help it. But if you are more than 50 years, damn sure, it is difficult. Anyway, there are on. Natural, these are all called the natural defensive mechanisms. Right. Next.
Sorry, sir, one
Yeah,
last
it is
question
moving.
to
Yeah.
both of you, Professor Manas sir and to you. Sir, ROS is a qualitative phenomenon or quantitative phenomenon? What is your opinion? In your opinion, whether ROS effect is more qualitative or quantitative for the cell perspective? Means if this is a concentration, ROS can be considered as quantitative, or it is with respect to lethal gene, it is qualitative one. What is your opinion?
It is both, I think.
It is both actually, depending on the how much it is generated. Somebody asked, so we have huge enzymes that kind of superoxide dismutase and all kind of free radical, basically whatever ROS means free radical reduce. So we have that. When it is breaching means quantity is much more, then the digester is happening like the one digester effect is the necrosis. What is happening there? Huge inflammation happens and body is basically not tolerable, so their damage happens. Okay, so both qualitative as well as the quantitative.
It is more quantitative one instead of qualitative.
No. See, it is bearable one certain, and if it is there, it will kill everything. Okay, and then huge inflammation happens. So the bad thing about the ROS is the inflammation happens because it major pathway of death is that necrosis if it's breach the medium. So that's why generally drug should not induce the necrosis because it will kill your own body, not because of drug. This is kill, basically damage everything. Okay, the inflammation huge come, immune response will come. So that's why the apoptotic like nanoparticle which induce apoptosis, that drug will be much better for the human health than the necrosis, because unnecessary immune system will be activated.
Thank you, Professor, for the nice explanation. Right. Yeah. Go back. I think video is working. I have seen it is moving. Just click it on the control. No, leave it. No problem. Okay, we'll go to the next one. Go to the next one. This is the simple assay for the cell migrations. We mimic the wound in the in vitro conditions. And we make the scratches. It is earlier we used to make the scratches, the manual scratches. So in the manual scratching system, there is the edge is not clear. But we have the silicon, blocks, silicon blocks, which is non-irritative, non-reactive to the cells, and make it and the cells grow. These are all the fibroblast junctions. And time lapse studies 12 hours, 24 hours, 48 hours, whatever it is, and see that how cells will move from this edge to here, this edge to here. This is the strategy in the control in the 12 hours. In the 24 hours, of course, cells are moved here. In the 24 hours, more cells are there. Whereas in the nanoceuticals, what I did is the silver nanoparticles conjugated with polyphenol curcumin and added the same phenomena in the 0 hours, 12 hours. Just to compare this to this, cells are of course better, movement is of course better. In the 24 hours, it is almost healed. That's right? Because in the 12 hours, this portion is not healed because it is a H cell phenomena. It is called that there is one phenomena, H cells are very resistant. Professor mentioned the one word, I don't know how many people, how many students and people are captured or listened that one, it is the cellular senescence. That phenomena is working, and the H cells are very resistant to move because of the cellular phenomena, cellular senescent phenomena. Next. Here also, I tried the TGF beta is the most important. What I did is the wild type cells are knocked down. The TGF beta is knocked down with the one technique, very simple technique, it is CRISPR based assay. We knock down completely the TGF beta, and knock down TGF beta cells are labeled with the green fluorescent protein, GFP. So we have two cells. One is the wild cells, it is the normal cells. One cell is the TGF beta minus. We co-cultured the both. Since it is we cannot see and I judge which is the knockdown cells and which is the wild cells. For that, labeled with the GFP proteins, then we co-cultured it. And you just try this one. This is the video. This one. See? Yeah. You see, the gray color is the wild cells with the TGF beta plus. That means there is not knockdown. The green color is the knockout cells, GFP labeled cells. If you see, once again, please, this you see clearly observe it. Potential of the migration of the wild cells are more than the green cells. That means TGF beta minus, 100% this one. That means I try to prove it. TGF beta is more important for the fibroblast migrations. One thing. Second thing is the confocal. Professor mentioned very well that what is the strategy, what is the cellular architecture of that one. We observed that TGF beta minus, where the skeleton architecture is less than the ordinary TGF M5 SS, the fibroblast cells with the plus cells. We published in 2018 Materials Today Proceedings, this work. Yeah. Next one. The photothermal reagent that I am not going to explain how the helps in the cancer biology. Theranostic. You may be knowing that one phenomenon is there, it is called theranostic, the nanoparticles with the diagnostic as well as the therapeutic molecules. Especially this being used in the cancer biology. Yeah, next. Cardiotoxicity, recently I started with this one, and the cardiac cellular senescence reversal mechanism by the nanoparticles. Cardiac cell cellular mechanism, cardiac cells, cardiomyocytes, this is the important cell for the heart health. So I started working on the how cellular senescence, cardiac cell senescence increases the fibrosis that leads to the heart attack. That phenomenon hypothesis, I started working on it. Yeah. Next. Transition metal. It is Xens. It is a highly physics one for the electricity and other things they use. This is the layered sheet metals, layered sheet, the nanoparticle sheets, and the in between sheets, it is a slippery. Due to the external simulation, that slippery potential will increases or decreases, so that the interactions or exfoliations through the etching, that means external molecules, have the more advantages to produce the energy. It is not for the biology, but my interest is here, if you add some photothermal reagents between the during the etching phenomenon, intercalcation and exfoliations, if we have the sliding mechanism is difference, if it goes to the cell, if the sliding mechanism is different, if you have the drug in this sliding portion of the nanoparticles, if it is captured by the cancer cells, because cancer cell has the foliate receptors to target the specific cancers by the foliate receptor mechanism from this exfoliation mechanisms. This is my interest, but I have to, it's only hypothesis, just only hypothesis, so it to do the work. Next one, Zenas one, I think. Yeah, next. Yeah, these are the life science mechanisms. How the vaccines are helping in the diagnostic, therapy, toxicity, and antibacterial, environmental, and biosensing. All these are there. So major being used in the imaging techniques, CT MRI, CT scan, and implants, drug delivery, in vitro, in vivo, membrane potentials, and all things we can use it. This once again the hypothesis. Yeah. Next, genus nanoparticles. Excellent nanoparticles. If you have the one particles which is nanoparticle, when it enters to the bloodstream, the plasma, which has the clumping mechanisms. If there is a clumping mechanism there, the desired effect of the nanoparticle will not be seen inside the cell. In order to overcome that clumping or aggregations, it is called aggregation mechanisms, we can use the genus nanoparticles. That means it is the clumped nanoparticles. If you inject that one through the nozzles with electrical or other stimulation, external stimulation, this clumped nanoparticle will be disintegrated into individual nanoparticles. The best one is the sonication, but sonication once again has the problem of the destroying the entire cell. We cannot use that one. So any other particles, any other mechanism or any other instruments or any other natural or synthetic chemical can be used to disintegrate the nanoparticle inside the cell. That one has to identify. Next. As the magnetic therapy for the wound healing, the muscle cells, if you give the magnetic and optical stimulation to the Petri plate, for example, if a streptomycin is there, which have the desired effect on the inflammations, but if you give the direct optical or magnetic field to the cell, once again it is a problematic. So that desired effect will not be there. So in order to have the better efficiency or efficacy of the streptomycins through the magnetic stimulation, because in the magnetic stimulation, cancer people, cancer clinician, cancer doctors, they use the magnetisms inside the cells to kill it. But it has the desired and undesired effect both. In order to have the desired effect of the drug for the external physical stimulations, can we have the more cellular inclination or cellular proliferations? Any other mechanism is there or not? So one has to identify. These are all the hypotheses. One can work in the future angle how the physical and biological effect have the beneficial efficacy towards the cell. Next. So these are all the magnetic hydroxyapatite particle is used completely in the bone regenerations. This is the most important. The bone has the two principal cells, the osteoblast and osteoclast. Osteoblast is good for the bone regeneration. Osteoclast, which consumes the osteoblast, which disintegrate the bone. The best problematic and issue is the nowadays is the menopause osteoporosis. So what we have done is we encapsulated aloin, the natural aloin. It is isolated from the aloe vera, and that aloin has the what you call balancing effect on both on the osteoblast and osteoclast. So we proved the long back, some 10 years before we proved on the aloin, it is anionic forms. Now what we have done is we encapsulated aloin with the magnetic hydroxyapatite. Magnetic hydroxyapatite is the filling agent for the bone. The dental implant and other things, they use it. And it is a non-reactive to the cells. So wanted to have this work already we started it in our lab. So what is the effect of aloin incorporated hydroxyapatite for the bone regenerations? Yeah, next. This is how the protocols, it is called SOP, Standard Operating Protocols for the bone regeneration by using the electrospinning mechanisms with the tendon and ligament regenerations with aloin. Aloin is a molecule, we already proved that it is working excellent in the bone regeneration. Then we wanted to have the what you call in situ extracted tissues, decellularized extracellular matrix with the coaxial spinning mechanism, with the 3D printing, 3D bioprinting mechanisms, and prepare the scaffold-free 3D spheroid models. And normally cell cultures are in the 2D dimensions. Nowadays the spheroid models are available. So we prepared the scaffold 3D models of that one and inject directly into the rat or a mouse or a rabbit models and inject it and see the ligament injury can be reversed or not. Next. And cytoskeleton decay is another important where when the time increases and certain degeneration mechanisms, there will be a complete decay of the cytoskeleton, actin, vimentin, and all other things will be there. That in simple principle, cellular architecture mechanism is destroyed in some certain diseases, especially the wound healing and some other disease also. So we wanted to have what is the efficacy of nanoparticles to increase the prevent the cellular cytoskeleton decay. This is that assay. Next one. Tendon healing, I mentioned earlier. This is how we prepare the 3D bioprinting mechanisms, and we made the channelizations. And our aim is I have some collaboration with the engineering department, bioengineering department, where they fabricated microfluidic chambers. And we can see the entire mechanism in the one slide. So many people are working the lab on chip and cell on chip and all the things are there. So these are all the simple mechanism to study in our control. Tell me. Next. This is how we prepare the different microfluidic chambers and how the nanoparticles and other things have the differential effect on the magnetic bonds, and model animal is E. coli or SRS and other things. First we have to identify the cellular toxicity of that one. This is the best example to study it. Then cellular, come to the cell models, right? Next. Lab on chip models for the nanomedicines, how individual organs can be developed into the organs through the spheroid models, artificial heart, artificial lung, artificial kidney, and artificial blood vessels. Everything can be done with the mechanism of the spheroid models with the microfluidic chambers. Next. Yes. Womb to tomb trajectory of the path. So this is the one project I am running with one university in Karnataka. Japan's growth period, economic zone, and genetic zone is highest. Next. And in India it is not so. There is a large difference between Japan to India in all three phases of the life. This is the reason. Next. 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 No, go back. So our lifespan is around 69. This is ICMR declared. Japan's is 92 years. So almost 90% of public will live 92 years. Whereas we 67, 70, nowadays just improved. What is the reason? We both are, our RNA, DNA, protein, everything is same. But what is the reason, that one? So there is one global project is going on where India is a recent signatory for that. It is called Future Child Crew. Future Child Crew. The global project, America, you may be knowing the Quad project, Quad, QUAD. So in that project, America, Japan, Australia, India, UK, Poland, and New Zealand, they are the signatories. And the same project I am running with the Nitya University. So yes, next one. The results are encouraging. Good water, good health, good air, everything is good. But problem is there is a cord blood, cord blood which connects the womb to the child. If you when I am doing that and I did the cord blood array, severe problems in that cord blood. But good child, no problem. Healthy child, good delivery. Of course, cesarean, this is the fashion of the nowadays. That is, forget it. But cord blood, if you see the cord blood array, severe disformity in the gene. That is the reason we want to know. We are going to, of course, this is not yet over. Next year, if the project is going, we want to have the second phase of that project with the medical college. I am coming to Pune, sir, for the DPU Medical College. That time I will discuss that result with you. Next. These are all the different, go back. These are all the different publications, important publication from myself and the global citations in the GCI. Next one. And next. This is the cover page. The two cover page we got from the recent. One is ACS biomaterial, one is colloids and surfaces. The cover page we got. Next. Yes. Finally, सबका साथ सबका विकास विश्वास प्रयास और जिज्ञास होना चाहिए. These are all my collaborators, including Atmiya. Probably NCCS is the next collaborator. Whenever I go, I increases the logo. Some space are here and some space is here. Let us join together. Thank you. I think over. Yeah. Yes. No, these are all, forget it, no problem. This is how fast, once again I thanks, this is the fourth time I am thanking Professor Manas to cut short the flow of the questions for the students. What is cell, what is protein, what is signal transduction, that and all he completed. So try to link my slides with his slides and develop or ignite a new hypothesis for the research. That is for you and for the university. Both it is better. Thank you.
See, this is the beauty of science. Concepts and applications, they go hand in hand. Truly, sir, that was an enlightening talk on the strategic criteria regarding nanomaterials in biomedicine. Now I would like to request Dr. Krishna Joshi, our HOD, to present sir with a memento and a certificate. Yes, who was clapping? Very nice. Please, keep clapping, everyone. Thank you, sir. Thank you, ma'am. Once again, I thank all the dignitaries, faculty members, and all the participants and the students who are attending over here for putting together such a beautiful, wonderful event. Especially thank the coordinators, Shivani Tank ma'am, Mosami Das ma'am. Now let us move for something exciting, okay, which is Chai Pe Charcha. Actually, I am presuming it is time for the break since some time. So the participants who have registered, the students who have registered, kindly move to the lobby outside this auditorium for Chai Pe Charcha, which is high tea. And any other students, if they are here, kindly go ahead with your regular schedule. And after, yeah,